About some of my PhD and postdoc work related to this extracellular matrix regulation for tissue engineering and regenerative medicine. So as the title suggests, my research is broadly focused on tissue engineering, which is, I'll just explain it for somebody who don't know the field. It is an interdisciplinary area which combines biomaterials, cells, and growth factors to develop 3D functional tissues to replace, restore, or regenerate an entire organ or injured tissue. And this tissue engineering field, which is relatively new, has made tremendous progress over the years in different facets of engineering, especially in engineering cells, because we have the advent of this induced pluripotent stem cells or adult stem cells, and we have several gene editing tools, and we have found different ways to modify the chemistry of the biomaterials which we use, and also we have found different strategies to deliver growth factors and different ways to modify the material properties, such as in the topography of the material, or the wettability or the stiffness of the material. And also we have made tremendous progress in terms of newer biofabrication tools, especially 3D bioprinting or different self-assembly process, and also like decellularizing an entire organ so that you can transplant it in patients who have like a disease, like an organ failure. So although like there is a tremendous progress in all these small areas of tissue engineering, there is still an unmet clinical needs which has to be addressed. And so my research has been broadly focused on tackling many of these challenges which are currently existing, especially in terms of developing a mimic which can like both structurally and functionally recapitulate the organ functions. So so my lab here in the institute, which just started, so we will focus mainly on different facets of this tissue engineering and regenerative medicine, where some of the areas which I'll be interested in is extracellular matrix manipulation to tune the properties of this matrix to instruct the cells to perform a particular function, and also to develop some non-invasive method to regenerate injured tissue or organ, especially by means of injectable hydrogel systems, and also to develop this 3D tissue or organ equivalents using bioprinting techniques or other fabrication techniques, and to understand neuroimmunomodulation using some of the physical factors like electric stimulation or magnetic stimulation. And also I'll work on disease on chip models, especially to develop this miniaturized microphysiological platforms that can be used to study diseases, and also that can be used by these pharmaceutical companies to screen some of the drugs which they develop. So these are some of the areas which I'll be working on in my lab here at the department. So if any one of you are interested in working in similar fields, you can reach out to me for your like final year thesis or something. So I'm just throwing out these ideas to you. With that, I'll move on to my research presentation. So I thought like I will present two of my research work from my postdoctoral stint. So the first work which I'm going to present today is on vascular calcification model, which this work is conducted in University of Minnesota during my second postdoctoral training. So the background for this one is that So vascular calcification is one of the major contributor of cardiovascular diseases. So if you look at our tissues in the body, that can be broadly classified into two types. So one is the soft tissues, such as the brain, the blood vessels, the lungs, the liver, and so on. And also the hard tissue, which includes the bone, the enamel, and something like that. So the hard tissues are like very highly mineralized or calcified, which is important for their function. For example, the enamel, which protects the tooth from grinding or some acid attack or the bacterial infection, and the pressure induced while you are chewing and all that. So that is important for its function. And similarly, if you see the mineralized nature of our bone, that is quite important for it to give the structure and form for your body and also for locomotion. So hard tissue calcification is something which is very desirable and important for them to perform its function, whereas soft tissue calcification is something which is detrimental and life threatening. So if you look at calcification happening in the blood vessel, that can lead to like severe cardiovascular diseases. Especially, it is known that some of the statistics show that it can be present in most of the individuals who are like above 70 years old. So that is one of the main reason for arterial hardening, and that can lead to like severe complications later on, including like reduced distensibility and compliance. So if you look at why is it important to understand vascular calcification, is because if you understand that, then only you can create treatment for that. For example, if you have a highly calcified blood vessel, then it is very difficult to penetrate a guide wire through it or the catheter through it, or if you want uh, to to uh, to introduce a stent there, or some re-entry devices, it will be extremely difficult because of this calcification. So understanding this vascular calcification or developing an in vitro model for this calcification is highly important, so that you can find out some treatment methodology to mitigate this calcification process. So here I have shown some not the schematic, some pictorial representations here. So this is an histology image of the cross section of an artery which is calcified, and you see this pink area here that shows that they are not calcified; they are the normal tissue. Whereas you see this dark regions here, which is the calcific deposits present, and the stain which they have used, which is von Cosa, that specifically go and bind to these calcific deposits, and that's what you see here. And similarly, if you see the X-ray of a person who has this kidney failure, because in patients who have kidney failure, they have higher concentration of this calcium and phosphate in their serum, and that can cause the deposition of these minerals in these blood vessels, and that's what you see, this brighter, dense calcific deposits in these blood vessels, which can cause severe cardiovascular issues later on. This is other causes for this vascular calcification. So one main cause for this calcification is this end stage renal disease, which is the kidney failure, and also patients with diabetes, they are prone to have vascular calcification. Similarly, people who are aging, they also tend to have like this stiffening and calcification of the blood vessels. So there are several reasons for it to happen. One is, as I mentioned before, it is because of this increased calcium and phosphate level in the serum, and also it can be due to some of the inhibitors which are inactive, especially some of the proteins which can act as mineral inhibitors. They lose its activity, especially osteopondin or fetuin or pyrophosphate. So those are like the other reasons for vascular calcification. Similarly, the vascular smooth muscle cells, which is present in the blood vessels, they can transdifferentiate to bone-like cells. So they tend to secrete more of these minerals, and that's how they they form this calcified deposits there. So all these reasons contribute to the development of this vascular calcification. However, all these existing in vitro models, they don't mimic the complexity of this native calcified arteries. Especially if you look at the models which is currently available, that is like they usually isolate the smooth muscle cells from the arteries, and they grow it as a monolayer culture. And then they induce this calcification to happen, which is like a sparse deposit of calcification happening in the culture plates, which no way mimic the architecture of this calcific deposits present in the native arteries. And similarly, there are other models where they usually section the artery or the artery of an animal, especially rat or mice, and they culture that to induce calcification. Again, they cannot be used for device testing, because you really need a graft with sufficient length and diameter so that you can introduce some of the devices to treat these calcification. So that's why they severely lack the structural, morphological, and functional attributes of the native calcified artery. Similarly, they are not physiologically relevant, because they cannot be used for device testing because of its because they don't have that appropriate dimension. So that's why we came up with a method to develop a tubular graft which can be calcified so that it can be used by these device testing companies or by the pharmaceutical companies to find out ways or identify ways to mitigate vascular calcification. So the method we use is like it's a protocol where we use this fibroblast from our skin cells. We take this fibroblast, we entrap them in a three-dimensional fibrin network, and then what we do is like we inject them in a tubular mold with the dimension which we require, and then we culture them for like 7 weeks. What happens is these cells are highly contractile, and these cells they undergo compaction, and they circumferentially align these matrices and remodel the matrix and synthesize the matrix in such a way that you will end up with a tubular scaffold with the cells entrapped within them. So we usually don't want the cells in there, because that will be difficult when we are thinking about transplantation later in the body because of immunological reactions. So we later remove the cells, so you only have the cell secreted extracellular matrix in there. So that's how we create this off-the-shelf biologically engineered graft. And this is an example showing the graft which we synthesize using this method, where we can have the extracellular matrix purely secreted by these cells. And this is the histology image showing the cross section of these grafts with the cells in there, and later on we decellularize them because we don't want the cells there. We only want the structural support for further application. So what we do is like we decellularize them, and you can see that you get this graft with this extracellular matrix, which has like a lot of the properties similar to that of the native artery, because native artery also is composed mainly of collagen and other proteins and other ECM related proteins. So all those proteins can be seen in these matrix, which is synthesized by these cells. So then what we did is like we induce this artificial calcification in these vascular grafts. So we use a certain concentration of this calcium and the phosphate in these in the culture medium in which we are growing these grafts. And we also add some calcification inhibitors. Like for example, pyrophosphate is something which is present in the serum which can inhibit the calcification. So what we do is like we add a particular concentration of alkaline phosphatase which can inhibit this pyrophosphate. Along with that, we wanted to test different serum concentration because serum has certain level of proteins which can help in the controlled deposition of this mineral crystals. So so we were able to achieve several of the key aspects of the native vascular grafts along with the calcification. So the first and foremost thing which we were able to achieve, which is important for device testing, is the tubular geometry. We were able to get these tubes of desired diameter and length using this approach. And we also tested different serum concentration. And we were also able to achieve this transmural diffuse calcification on these graphs. We also found that these calcium deposits were incorporated within these extracellular matrix fibers in an aligned fashion, and that alignment was noticed by image analysis tools and by a technique, fast Fourier transform technique, where we can identify the pixels which are oriented along the direction of this calcific deposition. So all this clearly shows that we were successful in mimicking the tubular geometry, the diffuse
Excuse me, ma'am.
calcification present. Yeah.
Your screen isn't visible.
Oh, it's not visible now.
No.
Did it go now or?
Yeah, it went maybe a minute ago.
Okay, just a second. Let me see. So can you see now?
Yes, ma'am.
Okay, all right. Yeah, please alert me if you don't see the screen, okay? Because I have no clue if you are not, if you are able to see or not. So please do let me know. All right. Okay. So moving on. So we also did some ultrastructural characterization to see how these calcific deposits look, and are we able to replicate the exact calcific deposits which you would find in the native calcified artery. So we did this SEM imaging, and what we noticed is that these calcific deposits were spherical in nature and they were poorly crystalline, which is exactly similar to what you would see in the native artery. So shows that to some extent we were able to mimic the exact morphology of the calcium deposits in our artificial artery. We also did some chemical analysis to prove that, okay, we were able to get these calcific deposits incorporated within this collagen matrix, and this is the FTIR analysis showing that that these calcific deposits were in turn incorporated in the collagen matrix. We also did some thermogravimetric analysis to measure the mineral content in there. And here it shows that with different serum concentration, we were able to, achieve a higher calcific deposition. In our case, with the 10%, we were able to get like almost 60% mineral deposits in the calcified grafts. So one of the main thing for a device testing study is the mechanical properties, because once the calcified artery or the artery gets calcified, their compliance like reduces tremendously, and identifying that is important, because then you will know how much. So when you are inserting a guide wire or when you are inserting a balloon stent, you will know the extent to which you need to dilate it. So we also did some mechanical testing for this. So the first thing we did was like the uniaxial stretching of these grafts. Surprisingly, we didn't see any difference with the non-mineralized and the mineralized grafts, because they both were showing similar tensile properties, especially the ultimate tensile strength as well as the tangent modulus. So then what we did was we looked at the compliance of this matrix, of these tubes. There we we were like checking the cyclic pressurization of these graphs, and there we saw like there was a tremendous decrease in the compliance, almost, which we calculated to be 16-fold reduction in the compliance, clearly showing that this calcification has reduced the distensibility of these grafts, which is similar to what you would see in the native calcified arteries. So they become so stiff that you won't be able to dilate these calcified grafts. So all this clearly shows that we were able to achieve a similar mechanical and the structural property of a calcified artery in the humans. So we also wanted to find out whether can this be used to used as a screening method to find out the concentration of a drug to inhibit this calcification. So we took a particular drug, which is this hexazodium phytate, which is currently in clinical trial. So we wanted to check whether can we use this to see whether that can inhibit this calcification. So we used a particular calcification which they were already using it in kidney failed patients. So what we saw is that using this compound, we were not able to completely inhibit the calcification, but we were able to induce significant reduction in the calcification deposits there. So clearly shows that such models can be used to screen some of the drugs which is currently in development for the treatment of vascular calcification. So in summary, what we have done is that we have developed a biologically engineered calcified artery which is completely off the shelf, and it has this tubular geometry and the matrix composition exactly similar to that of the native artery. And we were also able to induce this calcific deposition within these matrix, and then we found that the mineral composition and the morphology is approximating that of the native calcified artery, and it has this close association with the collagen fibrils, and we were able to achieve this distensibility and compliance similar to that of the native calcified artery. So all this clearly shows its potential for testing newer devices or testing new drugs which is currently under screening for the treatment of this vascular calcification. So this is one study where the calcification in the soft tissue, which is highly undesirable, but calcification in the hard tissue is something which is very much desirable, because that is important for them to perform its function. So I will move on to another study where I'll show how much the calcification is important for the matrix or for the tissue to perform its function. So that is, for that, the model tissue is bone. So the structure of bone is quite fascinating, because the bone is like a very highly calcified structure, where these cells, if you see the bone cells, which is osteocytes, they are cemented within these calcified structures, which you cannot like mimic in a lab setting, because these are like the cements, and these cells are like entrapped within them, but these cells survive these highly mineralized microenvironment. So in addition to that, they have a lot of this highly interconnected vascular networks, which is important for the nutrient supply for all these cells. And also they have this nerves present, this innervation present, that is also important to transmit the signals in and out of the tissue. So like existing scaffolds which mimics this bone microenvironment is very simplistic. For example, there is a lot of synthetic ceramics which is currently available as bone substitute, which no way mimic the cellular composition or the vascularization of the native bone. And they have the mechanical properties or stiffness way higher than that of the native bone. And there are like hydrogels present, which is like extremely soft, and they don't perform the mechanical functions as that of the native bone. So only alternate strategy is to use autografts or allografts. So autografts are the bone which is the grafts which is taken from your own body, or allografts is something which you are taking from a donor. So those are the most desirable one, because that is like what the nature has created, which is a highly complex structure, whereas the one which is there in the existing in the existing literature, they are like so simplistic that they don't mimic many of these aspects present in the native bone. So why does it work? The autologous bone or our native bone always works. Why is it? Because it is highly vascularized. Because it's vascularized, it can immediately connect with the tissue, and then it can, you know, constantly supply the nutrients and oxygen to the tissue, so to the cells especially, so that they don't starve and die. They also have this resident cells, which is osteocytes. So that is like 90% of the cells are osteocytes in there. So these osteocytes are like entrapped within these mineralized microenvironment, and they kind of send out the signals to the outside environment, so that the cells from the outside the bone, such as that of the osteoclast, they can like home to this particular area, and they can like constantly model and remodel the whole tissue. So that's very important. So in addition to that, it also needs the cell microenvironment, which is mainly mineralized, and also it has collagen in there, because the collagen is very important for the cells to grow, because they have the receptors for the cells to attach to them. So that's very important. So all these things make autologous bone the ideal one, and or the gold standard one, and it is very hard to mimic that in an in vitro scenario, or in a laboratory. So so that's why we took that as a design criteria, to make a microenvironment which exactly mimic the native microenvironment. So which includes the 3D vasculature. It has the resident cells, especially the osteocytes, which is derived from the mesenchymal stem cells. So these stem cells go through different stages, from being a pre-osteoblast to a mature osteoblast, and then they convert to a bone forming osteocytes, which is entrapped within this mineralized microenvironment. And similarly, it has other components in the microenvironment. So one is this nanoscale mineralization. So the one fascinating thing about these minerals present in these bone microenvironment is that they are nano-sized apatites, and these apatites are deposited in such a way that they are aligned along the long fibers of this collagen. So and then they are all present in the all present surrounding these cells, and you have this collagen, which is like collagen and other non-collagenous molecules, which is also important and contribute to this microenvironment. So what we did is we developed a technology where you can enwrap the cells, especially the progenitor cell, like the mesenchymal stem cells, in a three-dimensional collagen fiber matrix, and then you can induce controlled deposition of these calcium phosphate into these into these fibers, using a technique called polymer-induced liquid precursor process. So using this process, we were able to achieve like rapid mineralization of these extracellular matrix just in 3 days, with the cells encapsulated within them. So we characterized the matrix first to see like we were able to achieve that mineralization or not. And here this is an example where it shows the non-mineralized collagen fibers with this debanding there. So you can see that once you induce this deposition of these mineral crystals, you can see all these dense apatite crystals depositing along these fibers. And if you like do a high resolution TEM imaging, you can see these individual hydroxyapatite crystals, which are like, they are this nano-sized apatite crystals, which are aligned along the direction of the collagen fibers. So we also did some chemical analysis there to see whether we are able to incorporate these calcium phosphate crystals within these collagen fibers, and we found that the mineral to matrix ratio achieved using this method was almost similar to that of what you can see in the native bone. So which is pretty accurate. So it clearly shows that we were able to achieve a bone-like mineralization. In addition to that, we were wondering, since we induced this rapid mineralization, is it detrimental to the cells or not? So we did viability analysis, and also we looked for the reactive oxygen species generation, because if there is any toxicity issues induced by this calcium phosphate, then that can kill the cells. So we saw that there was not much reduction in the viability in the cells which were entrapped in these calcified matrix. So this is just to show like how these fibers were mineralized. So you see the very light, these collagen fibers which were non-mineralized, whereas the dense apatites deposited onto these collagen fibers clearly shows that we were able to achieve this nanoscale apatite deposition in these microenvironment. So then we also looked at the three-dimensional architecture of these microenvironment with the cells entrapped within them. Here I wanted to point towards this the red region, which shows the mineral appetites present in there, and the blue region is the cells. So the cells, it clearly shows that the cells are cemented within these matrix, and they are like extending out some dendrite-like extension, which you would see in a native bone, where the osteocytes communicate with the neighboring osteocytes by sending out these filopodial-like extensions. We also were surprised to see that these cells, which were enwrapped within the stiff matrix, they were also showing many of the characteristic features of a native osteocyte, such as this lamina limitans like boundaries, and also lacuna like empty space within them. All this is the typical characteristics of the osteocyte. So this is very encouraging, because once when we are encapsulating a stem cell, which is a purely progenitor cell in a matrix which is very soft, such as collagen, and then we progressively trigger this mineralization, it transfers to become a stiff microenvironment, and the cells immediately sense that, and they also behave like a matured osteocyte, although they are like the stem cells. So we wanted to see like whether this is triggering some gene expression in these stem cells or not. So what we did was like we screened for many of the markers of mature osteoblast or osteocytes, and we saw that all these markers were expressed in a higher quantity in the cells which were exposed to this mineralized matrix. And we also compared it with a positive control, where the cells, these stem cells, were exposed to an osteoinductive medium, which is like a cocktail of some of the factors which can induce this differentiation to this osteocyte. So this clearly shows that without any growth factors or without any osteogenic supplements, these mineralized matrix alone could trigger the osteocyte-like transformation of this mesenchymal stem cells. So one important thing we notice here is that in our mineralized microenvironment, one particular protein, which is this osteoprotein, was significantly downregulated. So this is quite interesting, because if you look at this osteoprotein, we have to look at the ratio of one of the other protein, which is this rank ligand. So if you see that ratio is extremely high in the mineralized microenvironment. So what this suggests is that the cells which is in this mineralized microenvironment, they are secreting a lot of this remodeling proteins, which is important for the neighboring osteoclast cells to home to that particular region, and then undergo continuous remodeling. So this clearly shows that this matrix environment was triggering like a native bone-like phenotype, that the stem cells which was entrapped within these mineralized microenvironment, they were behaving exactly like that of the native bone cells. So one another criteria for a bone microenvironment is they should be highly vascularized. So we wanted to know, like, during like using our approach, can we induce this induce calcification in highly interconnected vascular networks, which is quite challenging. So we kind of tuned or optimized the protocol in such a way that we incorporated these endothelial cells within them, and then we triggered the mineralization. And as you can see here, even with mineralization, we were able to achieve these vascular networks in there. So one important thing we noticed here is that, so the stem cells which are surrounding these vascular networks, the cells which are like surrounding them, they were like expressing bone markers, especially one of the protein is RUNX2. So that was expressed more by the cells which is surrounded in a mineralized microenvironment, and the cells which are at close vicinity to these vascular networks, they were expressing something called the smooth muscle actin. So which is a pericyte marker, which is the one which supports the capillaries. So clearly shows that these stem cells can differentiate into two different lineage, like the one which is close to the mineralized microenvironment, they were expressing bone cell like phenotype, whereas the cells which are close to the vascular capillaries, they were expressing pericyte like phenotype. So we were able to achieve all this just by tweaking or modulating the microenvironment. So then we wanted to see, like, if we transplant it in an animal model or to a human, will that be like patterned and will that be stable for a long time? So what we did was, like, we took this non-mineralized microenvironment and this mineralized microenvironment. As you can see, this has this calcific deposits there. So we implanted it in nude mice, and we wanted to see, like, whether these tiny capillaries, they are still patent even after you implant them in the nude mice. And what we saw is that there was like the number of vessels which were present in the mineralized one was much higher than that of the non-mineralized one, which clearly shows that these mineralized microenvironment tend to protect these capillaries which were formed in these, and they were patterned even after 7 days of 7 days of implantation in these nude mice. As I was alluding to the fact that these bone matrix is also innervated because it has this neurons going in there, or the nervous tissue going in there. We wanted to see whether we can mimic that similarly. So we co-cultured this one of the neural cell line, and we were able to have them in there when we triggered this mineralization. So it clearly shows that we can have multiple cell types within that, and then we can encapsulate or cement them with this mineralized microenvironment. Um, so finally, we wanted to see, like, can this microenvironment, bone microenvironment, which we have developed, can that act similar to that of the native bone or not? Because then if that acts similar to that of the native bone, then we can use it to study many of the diseases which target the bone. Or if you want to understand some of the mechanisms involved, we can use this instead of isolating the native bone by itself. So one of the bone targeting disease is the prostate cancer. So in the late stage of prostate cancer, these cells can metastasize towards that of the bone, but the mechanism by which it happens, how it specifically metastasizes to that particular organ, is not really known. So what we wanted to see is, like, if you have this artificial bone-like microenvironment, can these prostate cancer cells sense it similar to that of the native bone or not? So what we did was, we implanted this non-mineralized construct and this mineralized microenvironment on either side of the nude mice. So on the On the right side is the mineralized one, and on the left side is the non-mineralized one. And after 24 hours of transplantation, we injected this prostate cancer cells there. And you can see these were like stained. So you can see that there was like a higher luminescence happening, expression on the regions where this mineralized microenvironment was transplanted, clearly showing that these cells are recognizing them like native bone, and that's why these microenvironment was providing like an enriched soil for these cells to proliferate and propagate. So this is quite promising. So this clearly shows that the microenvironment which we developed, which it closely resembles that of the native bone. So for that matter, you can use it to study many of the diseases which specifically target the bone. So the summary of this project is, again, so we are able to engineer a scaffold that shares almost many of the hallmarks of autologous bone, which includes the cells embedded in this highly mineralized microenvironment. We were able to make it like we can dynamically stiffen them, like from a very soft microenvironment to a very stiff microenvironment. And if we are entrapping these stem cells in there, we were able to transform them to like bone-like cell strictly by modifying the microenvironment. And this the mineralization profile that was like matching that of the native bone, because they have this nanoscale intrafibrillar and extrafibrillar mineralization, and we were able to induce osteogenesis without any osteogenic medium or without any growth factors, but purely by changing the matrix properties. And we were also able to incorporate vasculature and innervation in this microenvironment. So one thing, so the one disadvantage of this is that it still doesn't have the density or the alignment which you would see in the native bone. So this is more or less similar to that of the oven bone, or like the not the dense bone which you would see in like the long bone or something. So that is one of the disadvantages of our model, but perhaps we were able to achieve all other important hallmarks of the native bone through this model. So yeah, so it doesn't have the mechanical property similar to that of the compact bone. So it behaves like a spongy bone one would see near the bone marrow. So so that's pretty much the summary of that study. So as I told you, so this study was performed during my first postdoc in Oregon Health and Science University. So like I want to acknowledge all my postdoc supervisors, both in University of Minnesota and Oregon Health and Science University, and also my PhD supervisor in Indian Institute of Science, and also the funding agencies who made this work possible. So thank you for your patient listening, and I'm happy to take any questions. I'm sorry that the slides went off sometime in the middle. I'm sorry for that glitch. Yeah.
Hello, ma'am. Good evening. This is Narendra. Am I audible
Yeah.
to you? Yeah.
Yeah, I can hear you.
This is regarding the vascularized bone scaffold. So that vascularity inside that bone scaffold, if that scaffold is implanted inside the body, would that would that be like those blood vessels you're talking about? Would they be ingrow into that scaffold, or just the uh, flow it allows, or just the flow of the matrix it allows without the ingrowth of that uh, those blood vessels? Actually, are they be going to ingrow inside that scaffold after the
Okay,
implant?
so in the after you implant, you're saying, right? Yeah. So one thing is that so there is a process called as anastomosis. Means you have the native arteries, the capillaries are coming branching out, right? So this one, So whenever you are implanting a material or a scaffold which has, if it has a intact vasculature or capillary, the native capillaries which is present in our body, that should there is something called as vasculogenesis. They sprout out, and they kind of go and join anastomose with that of the transplanted one. So this is an ideal state, it is supposed to happen. But most of the time, whatever tissue engineered constructs we make, because we are like artificially engineering these vessels, they are not even patent. By the time you transplant them, they kind of like, they don't even sprout, or they don't even anastomose with the host. So if they don't anastomose with the host, then there is no supply of nutrients, or there is no supply of diffusion of oxygen or anything happening. That's why many times these transplantation fails, because if you are using some artificial tissues or tissue engineered constructs, transplantation fails because there is no contact between the host vasculature and the transplanted vasculature. If that happens, then that fails completely. So this is one of the important criteria to keep in mind when you are designing a tissue engineered construct for transplantation. I
So
hope I answered your
yeah,
question.
yeah, you did. I hope. I think that that generally is a difficulty in the larger bone scaffolds, I think. Would that be
Right,
so?
right, exactly. Yeah.
You also talked about the implanting of those osteoblasts inside the that scaffold. So those osteoblasts, they are derived from the stem cells. You said it. Those stem cells, are
Right,
they derived from the patient itself, who in whom
right.
those
Yeah.
that scaffold is to be transplanted?
Right. So
And
that is one way. Yeah, that's good. Yeah, sorry. Yeah, that's one way. Like if you want to, so that good thing about mesenchymal. So I have used this mesenchymal stem cells. So you can like isolate these mesenchymal stem cells from the bone marrow of a patient, and then you can use that to be transplanted later. So you don't have any immunological rejection happening because it's taken from the same patient. But for this study, I have used the mesenchymal stem cells not from a patient. It's like we usually get it for research purpose. So you need to go through several ethical consideration if you want to isolate it from the patient. So we took the mesenchymal, isolated the mesenchymal stem cells. That is for the research purpose. So it's from the some normal donor. It's not from a, yeah.
okay,
Mhm.
so is that like we are growing those osteoblasts outside the body in vitro conditions, or like that, and then those osteoblasts, they are implanted into that scaffold, right?
Right. So, so here we are growing these stem cells within this scaffold, right? So they kind of mature within that. So what we have seen is like, so there are different ways to stimulate them to behave like osteoblasts. Like you treat them with several certain chemical cocktails so that they behave, they kind of slowly differentiate to become the osteoblast. So in this study, what we have seen is that even without doing that, if you have such a, if you just trigger calcium phosphate deposition in there, that itself can act as a cue for this to happen. For the differentiation to happen.
Okay, is there a possibility that while that growing up those osteoblasts outside the body, there may be that we cannot actually mimic the conditions those are going inside our body. So the time at which these growth hormones they are triggering the mechanism, all those stuff. So is there a possibility that a few of them may become maybe uh, cancerous, or may somehow affect the property, and when implanted into the body, they can be a kind of bigger problem like that? Or is there a possibility to get those osteoblasts blast from the bone inside from the patient, or like if that patient requires that scaffold, something might have happened to him, like he might have lost some sort of bone inside his body. So we would have that scrap from there, and we can take that scrap and take out the osteoblasts? Is there a possibility kind of that? Maybe it's kind of funny, but is there a possibility?
Yeah,
We could do.
that's a good thinking. The one thing is the osteoblast, the number of osteoblasts present. So, for example, if you break a bone and you are, I know, like nobody want to do that, but you got a fractured bone, you would say, but isolating it will be like a very difficult process because as I told you, you have to decalcify that entire thing to get out the osteoblast. So since it is like the if you are talking about an adult bone, most of there won't be any more osteoblasts. It would have matured to osteocyte. So you only will have osteocyte, and the number of osteocytes you could get will be very low. So then you have to isolate that and expand it in the lab again for several weeks before you can transplant them. So that's why everybody prefers the stem cells, because you can isolate stem cells and you can take the stem cells from the umbilical cord, and you can, like, there is stem cell banking, right? You can cryopreserve them so that later you can like take out that stem cell and then expand, and then later on you can like transplant it to the patient. So that's much more easier than taking so because if you are thinking about getting a bone from the patient, again, that itself you are creating an injury there, which is not desirable or which you don't want to do. But yeah, it's a good thinking.
Oh, am I troubling if I would ask a few more questions?
Yeah, go ahead.
So, so those cells, those osteoblasts, are how they are implanted into that completely formed scaffold inside it. So we are implanting deep inside that scaffold, right? So how are we doing that? Is there any specific method like?
So here in our study, how we did is that we take the hydrogel precursor, means it's like in a liquid form. So you mix the cell and then you trigger that fibrillogenesis to happen. So you have, when the scaffold is forming or these fibers are forming, the cells are already present in there. So you have this, if you have a three-dimensional structure with the fibers and the cells entrapped everywhere. So that's how you get like the homogeneous distribution of cells there, because you are starting from the starting itself, from the pre-gel solution, you have the cells in there. So you don't have to introduce. So if you are making scaffolds by other ways, say you are 3D printing a construct, or you are lyophilizing and making a porous structure, you cannot get the cells go all the way into the scaffold, right? That's why we do the self-assembly process, where you have the cells in there and then you trigger it to form the gel. So you have them entrapped.
Okay, that's a very good technology. So one last more question, please. If we are transplanting that scaffold that is made up of some other biomaterial inside the body, so like if there is a small fracture inside the body, the bone starts regenerating itself, and that goes fusing into the other part of the bone, and that joins them. So if we are implanting that scaffold inside the body, so would that bone be like fusing with that scaffold and make itself uniform, or we have to deal with the screws or kind of other things to get into that fixed place?
Yeah, so it depends on where you want to implant. For example, so if you are like, for example, you met with an accident and then you broke your skull. Say you broke your skull. The skull is something which it needs like very less mechanical support, right? So there you don't need to have additional fixtures. So you can just put the scaffold there and it can heal later on. But if you break your long bone or something, because you are continuously, still you have to walk and everything, right? So there you will have additional fixtures like a titanium screw or something to put that in place, so that until it regenerates, you have the support, mechanical support there. And later on, once it completely heals, you can remove that whatever is support structures you have put in there. So it depends on the location where you are doing. If it's like a non-load bearing location, like a craniofacial reconstruction is what you're doing, you don't need any fixtures. You can just you have to but there again it is an anatomically complex structure. There again the complexity is there because the structure is difficult to model. There you have to get the CAD design of that particular defect, and then you have to, like, using 3D printing or some molding technique, you have to make the exact structure because you need to fit that, because it's craniofacial bone. So there it's the challenge is different, whereas in the long bone the challenge is extremely difficult. It's like totally different compared to that.
Okay, that means that can fuse or that can regenerate into it after some time. Okay.
Yeah, exactly.
Thank you so much. Thank you so much. Thank you.
Ma'am, I had a question.
Yeah, go ahead.
From the first part of your talk, you had mentioned that you were using the calcified grafts for testing different drugs. So how important are the different cells, since we're not using cells in the graft, how important are those while we're testing drugs?
Okay,
Like
that's a good question. Yeah. Right. So having the cells is always important, because the cells are the culprits here, right? Because the cells are the one which is secreting these minerals. I was like mentioning vaguely that these endothelial cells and the smooth muscle cells which are present in these blood vessels, they are not supposed to secrete any minerals, but they do that when there is, in disease states like diabetes or renal disease or anything happens, these cells start to behave weird, then they secrete that. So inhibiting, so some of the drugs which you are like targeting specifically to that of the cells, so where like you wanted to inhibit the secretion of this calcific deposits, there it's important to have the cells in there. But again, why we didn't have cells in there is because for the long shelf life. Because if you want to send it out to a device testing company, they wanted to like leave it for 6 months or so. So if you have cells in there, it's difficult. Then you have to have the media in there, and then you have to have the appropriate culture environment for all that. So it adds the complexity. Nevertheless, in the process which we have made these grafts, we can reseed this endothelial cells or vascular smooth muscle cells later if you want to study some of the drugs which specifically target the secretion of this calcific deposits.
Okay, yes, got it. Thank you so much.
But it's a very good question.